Thank you very much for that introduction, and thank you very much for the organizers for inviting me to give a talk. So I was indeed a research fellow at the University of Tours from 2018 to 2019, and we liked it so much that we are still in Tours. So I do work for a company now based in the UK, but I work from home and I'm still based in Tours, and I intend on staying there. So this talk is a bit of a mix of my work, some of my work at the University of Tours, and some of my current work at Certara Simcyp. And although the methods are quite different, they are definitely linked, and I hope that that will be evident in this work. And so in the very first part, I'll just give a very brief introduction to relevant alternative non-animal methods for testing skin penetration of chemicals or compounds, particularly in cosmetic formulations. Then I'll talk about my work some of my work at Tours, where we investigated in vitro the skin penetration of cosmetic ingredients. And then in the last part is basically physiologically based pharmacokinetic modeling of skin penetration as well. So a cosmetic company that wants to put on the market a new formulation with ingredients has to provide a battery of toxicity tests to prove the safety of the formulation and of the active ingredient or ingredients that are in it. And we will be focusing on skin absorption or penetration, which is one of the tests that have to be done basically. The traditional method in the lab, in vitro, to investigate skin penetration from a pharmaceutical or a cosmetic formulation is to use ex vivo human skin. So generally this is skin that comes from abdominoplasties or breast skin or animal skin. And the animal, generally speaking, the porcine skin or pig skin is the skin that's closest to human skin. So the issue with ex vivo human skin is that it's not easy to obtain, or it's quite expensive. You have to have a relationship with a surgeon, for instance. You can also go through some companies which harvest it and store it properly and provide it, but that's very expensive. The issue with porcine skin for cosmetic companies is that since 2013, they cannot generate any safety data that has used any sort of animal tissues. Here this is a sort of a schematic of a France diffusion cell or a vertical diffusion cell. Essentially, if you have a piece of excised skin, you place it between a donor cell, a donor compartment, a donor cell, and a receptor cell, and you place inside the donor the formulation containing the ingredient for which you want to test skin penetration. And basically, because of the gradient in concentration between the donor and the receptor solution, you will have passive diffusion through your skin membrane. If you then analyze the amount of the compound of interest that has accumulated in the receptor, you can get an idea of the skin penetration of that compound. I'll come to that again later. Conversely, you can also test the barrier function of your skin membrane, so whether it's good at keeping things out of the skin and the body, etc. So as I mentioned, animal skin is problematic for cosmetic companies, and excised human skin is difficult to come by. And for the past 15 to 20 years, there's a whole industry in research and development of skin equivalents or reconstructed human skins. And the very basic way of doing this is to seed in multi-well plates skin cells, so keratinocytes, or maybe combinations of keratinocytes and fibroblasts, perhaps also other dermal cells, onto a substrate to expose these cells to a culture medium such that they will over time differentiate and proliferate. And then at some point you expose the top layer of the cells to the air, so it's called the air-liquid interface, and this will cause the top layers of the cells to cornify, and this will create a stratum corneum. So this is the upper layer of the skin that is essentially the physical barrier of our skin, preventing water from vapor from leaving the skin, and also foreign compounds from entering it. And so you have, after 10 days to 3 weeks, basically reconstructed human skin in your 12-well plates generally. So The issue with this is that it's quite expensive, or it's actually very expensive. If I remember well, one 12-well plate like this cost, at least a few years ago, on the order of 1,000 euros or something like that. And so we, at the University of Tours, we wanted to investigate, does the barrier function of these reconstructed human skins change if we store them, if we freeze them like we can freeze excised human skin, if we freeze them at different temperatures and for different durations. Oh, sorry, I forgot to mention, this is just a snapshot of different types of reconstructed human skins from basically the two main companies that produce these. As you can see here, the multi-layer structure of skin is reproduced basically with the stratum corneum at the top, the epidermis and the dermis. But I should say that this still remains much, much, much simpler than real human skin. And particularly, a compound will penetrate through this reconstructed skin much generally to a greater extent and at a greater rate than through real human skin. So what we did is we wanted to explore, can we store these things, these reconstructed human skins, and if we do store them, to what extent does it alter the barrier properties? So we used EpiSkin, which is an established and validated reconstructed human skin, and we stored it at different temperatures, minus 20, minus 80, minus 150 degrees, for either 1 week and 10 weeks, or 10 weeks. Then we did cryosection of these skins, and we we analyzed the skins through Raman spectroscopy. So Raman spectroscopy is a non-invasive spectroscopic technique which will provide a chemical signature of the substrate that you're investigating. In this case, of the stratum corneum of the reconstructed human skins. So each of these peaks in the Raman spectrum arises from the interaction of the laser with a specific component in the tissue. So it might be lipids, it may be proteins. There's a whole literature devoted to the assignment of spectroscopic signatures to different components of tissues, including human skin. You can also investigate the conformation of proteins and the status in which lipids are found, the organization of the lipids. So this essentially provides a semi-quantitative technique of investigating differences between the reconstructed human skins stored under different conditions. We conducted this Raman spectroscopy with the different episkins stored under different conditions, and then we analyzed the data using some chemometric methods. And essentially, this analysis shows us semi-quantitatively the differences between it shows us that there are differences between the tissue stored, for example, at minus 20 degrees for 1 week, that's the light blue, compared to minus 20 at 10 weeks, that's the dark blue, compared to fresh reconstructed human epidermis, so not stored under any sort of conditions. And so we compared the various conditions, and we can assign these differences to specific peaks, so to specific alterations, or to alterations of specific components of the tissues. And so again, this is a semi-quantitative way of demonstrating essentially that different storage conditions will affect the stratum corneum of these tissues in different ways. In parallel, we conducted more quantitative experiments, in vitro permeation experiments. So with tissues stored under the same conditions, we exposed them to a donor solution containing resorcinol, which we use as a model cosmetic ingredient. This is a very common chemical in a variety of dermatological and cosmetic formulations. And so the resorcinol absorbs into and penetrates through the tissue and into the receptor solution. We collect the receptor solution at predetermined times, analyze it by high-performance liquid chromatography. We convert the concentrations to cumulative masses, and basically the result is a cumulative amount profile of resorcinol in the receptor solution as a function of time. And then from this kind of data, you can obtain fluxes of the compound through skin, permeability coefficients. You can estimate partition and diffusion coefficients as well. So again, in parallel to the Raman spectroscopy experiments, we did these kinds of in vitro permeation experiments for the various reconstructed tissues that we have. Here in pink is the permeation, the cumulative amount of resorcinol through fresh episkin, so non-frozen episkin. And these are the cumulative amount profiles for the other storage conditions. So basically, this indicates that the permeation of resorcinol through the tissue increases as a function of the storage conditions above and beyond the non-stored, the non-frozen episkin. So basically, the barrier of episkin has been compromised. And we can then quantify, as I mentioned, the fluxes and permeability coefficients and the lag time to the steady state here. And so this, in complement to the spectroscopic measurements, allows us to quantify for this compound and for these episkins to what extent these storage conditions have compromised the barrier. And so, yeah, that's my first conclusion. So we use, these are relatively, especially Raman spectroscopy in this context is a still emerging technique, non-invasive technique, so we don't have to extract anything from the tissue, we don't have to alter the tissue itself to obtain very valuable data, and it complements more traditional in vitro skin permeation experiments that we conducted. Of course, for a much more full understanding of the effect of storage conditions, you would have to study a wider range of permeants of different physicochemical properties and also different formulations, because the formulation potentially affects the penetration of a compound through skin as well. And then I just wanted to finish by adding that although my STUDIUM fellowship ended in 2020, I've been continuing to work with Émilie, Frank Bonnier, and others from the group in Tours on various other publications that explain the use of, look at the use of Raman spectroscopy in this context, as well as reconstructed human skins to test different types of formulations. So it's really great to be able to still work with the group in Tours. Okay, so now I'm going to switch gears a little bit, and this is a transition to some of the work that I'm currently doing. So in 2019 and 2020, some US FDA authors published a couple of papers that made a big splash. Basically, they said they applied different sunscreen formulations, commercial formulations, in a clinical trial to some volunteers, and they found concentrations of the sunscreen actives in the systemic circulation that were above the threshold established by the FDA for safety. So this doesn't mean that we should stop using sunscreens, but this does mean that we have to investigate the safety and potential risk of some sunscreen ingredients much more. And the FDA also basically published a Federal Register or some type of guidance indicating for 12 sunscreen actives, for 12 UV filters, additional studies, and in particular additional in vitro permeations, clinical studies and in vitro permeation studies are needed, because these 12 filters are as of yet not considered safe, effective, and safe. And so the type of clinical trial that's recommended is a maximum usage trial. So that's where the investigators have to try and capture the maximum uptake of the active of interest into the systemic circulation from 4 different types of commercial formulations containing the active of interest. How should these trials be designed? How should the commercial formulations be selected? How should the doses be selected? Well, the investigators have to perform in vitro permeation tests with excised human cadaver skin in vertical diffusion cells, which I talked about previously, as justification for the formulations and the doses used in the clinical trial. There were a couple of other publications by the FDA where they presented in vitro permeation testing methods for sunscreen formulations. Again, in this context, in the same context. So they used human cadaver skin, but across these two publications, they also tested different types of tissues. So heat-separated epidermis is simply where the dermis is missing, so this epidermis has been removed from the dermis. And dermatome skin is where you have a full thickness skin with containing dermis. And they also compared finite and infinite doses, so different doses applied onto the skin in the vertical diffusion cells. They also characterized the formulations through Raman spectroscopy. One of the important characteristics that they show is the distribution of globule sizes within the formulation. So different formulations, creams, lotions, etc., will have different globule sizes. And this is one of the important formulation characteristics that affects or that impacts the release of the active from the formulation and absorption into the skin, etc. So at Certara, we asked ourselves, or maybe if I go back a minute, these are two publications. These are multiple experiments. Again, not cheap to conduct and not time-wise and cost-wise. And at Certara Simcyp, we asked ourselves, can the in vitro skin permeation of sunscreen actives be predicted using some kind of bottom-up mathematical modeling? Can we parameterize the mathematical model such that the effect of various experimental parameters can be captured? And if so, can we predict the experimental data, the in vitro skin permeation data accurately? That means in this general field of pharmacokinetics within twofold. And so we address this question using physiologically based pharmacokinetic modeling. I'll briefly explain what that is. Basically, the advantages of PBPK modeling or physiologically based pharmacokinetic modeling is that you integrate systems data, drug and formulation data, as well as trial design. Systems data is the system that you want to investigate. So it might be the organ within which a compound is absorbed and distributes, potentially metabolizes. It can be the tissues, it can be the whole body. The drug and formulation data are the physicochemical properties of the drug and the formulation characteristics. And the trial design data is the dose, the route, so in this case it's dermal, the populations, the type of skin, or the type of populations that you're investigating. And these data are integrated into mathematical model to conduct mechanistic, so either to predict the pharmacokinetics, so predict the absorption, distribution, potentially metabolism and elimination of the compound of interest, and also to do mechanistic in vitro, in vivo correlations and extrapolations. And in the end, you want to predict the pharmacokinetics of the drug or the compound of interest. PD stands for pharmacodynamics. That's the effect. So briefly speaking, the model consists of a large number, a large set of differential equations. Basically, what we're trying to do is to predict the concentration of a compound as a function of time in the organ of interest. It can be in the systemic circulation, it can be in the lung, it can be in the liver, and it can be in the skin. And these models contain a ton of physiological parameters, as I mentioned before, as well as drug-related parameters, etc. So coming to the skin model, so within the simulator that Simcyp develops, there is a skin module for dermal absorption, and this is a schematic of it. So basically, we simulate and predict the absorption from different kinds of formulations into the skin, into the stratum corneum, into the viable epidermis, etc. Each of these tissues basically is described physiologically accurately as can be from meta-analyses of the literature. And also, within these, each of these compartments, we have quantitative structure-activity relationships which predict diffusion and partition coefficients based on the physicochemical properties of the compound of interest. We can simulate different kinds of populations. So, for example, healthy North European skin populations, elderly skin, diseased skin, for example, psoriatic skin. All this, the difference between these is the physiological parameters of the skin that change. And also, we can simulate different body sites, basically, because the skin on the abdomen is not exactly the same as the skin on the forehead, for example. This was a schematic for in vivo. We have a similar model, which is for ex vivo or in vitro permeation predictions. Essentially, this model reproduces the same physiology, but instead of having the underlying subcutaneous tissue and muscle tissues, we have a receptor compartment. So basically we simulate this in vitro permeation experiment that I described earlier. This is just a screenshot of one of the screens where we basically parameterize this. So for example, we parameterize the skin type that we're simulating, the cell type, so whether it's a static cell or a flow-through cell, and also various parameters related to this receptor solution, and also the geometry of the fusion cell. Okay, so now coming back to the AFDA's in vitro permeation data that I presented earlier, and that we want to try and simulate and predict. Into our skin in vitro permeation module, we input the physicochemical properties of an active. So we chose one sunscreen active, oxybenzone. We input the formulation characteristics that were available in these publications, and also the in vitro permeation setup, so the dose, the type of membrane, which were also described in the publications. And we predict the various types of experiments, we simulate the various types of experiments that they conducted. Sometimes we have to optimize certain parameters, like certain diffusion partition coefficients, because although the model incorporates quantitative structure-activity relationships to predict those, they're not always entirely right, basically. So we have to optimize certain parameters. But we never just optimize and stop there and call it a day. No, we have to validate verify the optimization by simulating something else, which uses that optimized value to verify our optimization. And so, just briefly, as I mentioned, this in vitro permeation paper, they looked at, they applied sunscreen formulations containing oxybenzone to excised human skin. The points here are the different the data from the different skin replicates. The green line is the mean prediction, and the gray lines are the 95 and 50th percentiles of the prediction. As you can see in the data, there's some variability, but by and large, we simulate this experiment quite well. This was for dermatome skin, infinite dose. We had to optimize one coefficient, but we used that optimized value to predict to simulate another experiment that they conducted, and we do not so good in the early points, quite well in simulating the final cumulative amount. I should say though that for this particular experiment they had n equals 1, which is not very useful. So ideally most likely you would have a range in this data. And then we've simulated another experiment with heat separated epidermis and a finite dose, and again we simulate the experimental data satisfactorily. We did the same kind of exercise for another kind of formulation, which was a spray. So they investigated the in vitro penetration of oxybenzone from a sunscreen spray. And here there were other inputs that were needed because this was an alcoholic spray. Part of that formulation will evaporate. So this is called formulation metamorphosis, and we have to incorporate that into the model and the simulation in order to accurately predict the in vitro permeation profiles. So basically, what I showed here, is that we are able to predict the experimental data satisfactorily, meaning within twofold. What I didn't show here is that we can also predict the skin retention of the active. So we looked at the penetration through the skin. Sometimes for locally acting drugs, you want to know how much has been retained at a given location in the skin. And we're also capable of doing that. For complex formulations, you need also reliable input data. You need to know solubilities, evaporation profiles, if that's relevant, and rheological properties that we all can input into the model to simulate the formulation and penetration as reliably as possible. And so this kind of modeling, if it's verified and validated, can help to replace or guide the design of experiments, whether it's the in vitro experiments or later on clinical trials. It can also enable in vitro in vivo extrapolation, and it captures the variability of in vitro data or in vivo data. Also, as a general exercise, we do sensitivity analysis So this is to understand the impact of certain input parameters on the model output. It's very important in model development, verification, and validation. In general, PBPK modeling is increasingly, alongside in vitro data, becoming accepted for drug formulation development, optimization, safety assessment, and virtual drug bioequivalence. And there were recently a couple of reviews from the FDA detailing such case studies. So thank you very much for your attention, and I'm happy to address any questions.